Good morning, dear students. This is your teacher Neelam Helawat, and we will continue our chapter, that is chapter number 11, Biotechnology, Principles and the Processes. And dear student, this is our lecture number 2. I hope in the previous lecture, you've understood the contents that we have discussed in the previous lecture. In continuing the concept, like we had started with the concept of restriction enzymes. And now we have understood the restriction enzymes, they are very, very important. Isn't it? Because they help us to break the DNA component into pieces. Isn't it? So what they do? They help us to break the DNA component to pieces. So we can say restriction enzymes, they cut the DNA into pieces. Right? So let's come to the, in continuation of that topic, let's come to the next part, that is separation and isolation of the DNA fragments. Okay. Now we know, just now we've discussed, the DNA will be cut into pieces by the restriction enzymes. Now these DNA fragments, they can be separated by a technique which is called gel electrophoresis. Have you heard this term, dear students? आपने ये वर्ड सुना है कभी? Gel electrophoresis. Okay, the fragments which are divided by the restriction enzymes, they will be subject to the gel electrophoresis. Now, dear students, we know that DNA is a negatively charged molecule. Okay, DNA is a negatively charged molecule. So, if we apply electric field in a medium, then DNA will automatically move towards the anode. Okay, and the medium in which we will apply this electric field is a agarose gel. Okay, that is called agarose gel. Now this gel is obtained from the seaweeds. Okay, and the DNA fragments, they will separate out on this gel depending upon the size. So the smaller is the fragment, the further it will move. So the fragments which are smaller, they will come to the bottom. The molecules which are larger, they will stay on the top. Okay. So we can easily see, like if you can see, this is a device of gel electrophoresis. Here you will be putting the gel, and the gel is agarose gel which is obtained from the seaweeds. Then we'll connect it with the battery so that we can apply the current over it. We can apply the electric field. And because of the application of the electric field, the DNA will move towards the anode. Okay. So depending upon this, we can get this kind of structure. So the ones which are heavier, they will be here, right? This is on the top, and then slowly they will move down. So the smaller the fragments, the further they will move from the more distance they will travel, right? And these DNA fragments, as you can see here, these DNA fragments, they can be seen as bright orange color bands. Okay, when they are stained with ethidium bromide. Okay, so we cannot see the DNA fragments without staining them. So for visualizing the DNA, for making them visible, DNA must be stained with ethidium bromide. And when we stain them with ethidium bromide, then they will give a bright orange color when they are exposed to the UV radiations. Okay, so we have to expose them to UV radiations. We don't need to expose them to the visible light. So first, two things we have to keep in mind. First, we have to stain them, and second, we have to expose them to the UV radiations, because they will not be visible they will not be seen in the visible light, right? So that the DNA fragments, they will separate. As you can see here, they will separate out, like as I was talking about. So during this technique, which is called gel electrophoresis, the DNA fragments, they will separate out. The largest on the top and the smallest will move a more distance. They will move further, right? So these kinds of bands, they will be obtained. Now these separated bands of DNA, okay, these separated bands of DNA, they will be cut out from the agarose gel. अभी जो है आपके जो band separate out हुए हैं, इनको हम agarose gel से cut कर लेंगे. And this process of separating the DNA fragments is called elution. Okay, this is called elution. And these purified segments, they will be used to construct a recombinant DNA by joining them with cloning vectors. तो जो DNA हमने separate किया है फिर हम इसके combine करेंगे with the help of the cloning vectors, right? Is it okay, everyone? So this is how we separate and we isolate the DNA fragments, the desirable DNA fragments that we need, right? Now let's come to the concept of cloning vectors. This is a very important, and we come to the second point. Like as you can see in the previous slide, we have talked about three you can say tools of the recombinant DNA technology. So we have finished one, that is restriction enzymes, which are called the molecular seizures. Now we'll start with the cloning vectors. Okay, we'll start with the cloning vectors. Let's understand what are the cloning vectors. So cloning vectors, they are basically the DNA molecules that can carry a foreign DNA segment and can replicate inside the host cells. So cloning vectors जो हैं ये एक तरीके के DNA molecules हैं जो अपने अंदर किसी बाहरी DNA को incorporate कर सकते हैं, and when they are injected into the another cell they can easily multiply. Okay, so they can easily replicate into the host cells. Like we can have two examples here. One is the plasmids, another is the bacteriophages. Right? Plasmids, that is a circular extra chromosomal DNA of bacteria. As you can see here, in bacteria there are certain extra chromosomal DNA which is present and which is circular. So आपके bacterial cells में जो है कुछ extra DNA present होता है जो कि circular होता है, ठीक है, and that is called a plasmid. Now bacteriophages means the viruses which infect bacteria. जो viruses हैं जो bacteria को infect करते हैं, they also contain a DNA. You can see here they also contain DNA, and they are covered with a capsid protein. Okay, they are covered with capsid protein. So both these two, that is the plasmids and the bacteriophages, they are our cloning vectors. Okay, they are our cloning vectors. Now bacteriophages, you must have heard the viruses that infect bacteria. Now bacteriophages, they are present in large number in a cell. Okay, they are present in a high number in a cell. Means the number is very high in a cell. Okay, so in the bacterial cells the number is very high. Similarly, the plasmids also, they may be present 1 to 2 copies per cell, or they may have 15 to 100 copies per cell. So they are also present in large number, right? They can also present in large numbers. Now if we have to link a foreign DNA with a bacteriophage or a plasmid DNA, अगर हमें एक foreign DNA को plasmid में या bacteriophage में insert करना है, ठीक है, then it will multiply depending upon the number of plasmids which are present in the body of the host. So if that multiply, then its number will automatically become equals to the copy of the plasmid DNA. तो एक ऑर्गेनिज्म के अंदर जितना प्लाज्मिड डीएनए है उतनी ही कॉपीज आपकी रिकॉम्बिनेंट डीएनए की भी प्रेजेंट होंगी, राइट? बिकॉज व्हेन द क्लोनिंग वैक्टर विल मल्टीप्लाई इन द होस्ट, द लिंक्ड डीएनए विल ऑल्सो गेट मल्टीप्लाइड. सो अगर क्लोनिंग वैक्टर्स मल्टीप्लाई करेंगे तो जो डीएनए उनके साथ लिंक किया गया है, उनके साथ इंसर्ट किया गया है, दैट विल ऑल्सो मल्टीप्लाई इन अ लार्ज नंबर. ओके, yes. So vectors at present are used. Okay, they are used constantly in this manner so that they can easily be linked with the foreign DNA, and they can also help in the selection of recombinants from the non-recombinants. Okay, they also help us to. So they are engineered in a way that we can easily link the foreign DNA in these substances, and we can also identify the recombinants from the non-recombinants. Coming to the features of a cloning vector. See, there are certain features that are required to facilitate the cloning into a vector. अगर हम चाहते हैं कि एक particular vector के अंदर हमारी gene multiply होनी चाहिए, so we need to have certain factors. First of all, the cloning vector should have an origin of replication, which is called the ORI. As you can see in the diagram, there is an ORI. Okay, so this is the origin of replication. Second is it should have selectable markers, means it should have marker gene. ठीक है, उसके अंदर जो है वो मार्कर जीन्स प्रेजेंट होनी चाहिए जिससे कि हम उसको आसानी से डिस्टिंग्विश कर सकें. ओके. सेकेंडली इट शुड हैव अ क्लोनिंग साइट्स. मतलब क्लोनिंग साइट मीन्स ऐसी जगह होनी चाहिए जहां पर अगर हम अपना डीएनए इंसर्ट करें तो वो ईजिली मल्टीप्लाई हो सके. ओके. सो फोर्थ, vectors for cloning genes in plants and animals. And the fourth most important, that we should also have vectors for cloning the genes in plants and animals. So 4 things must be there. First, there should be an origin of replication. There should be the selectable markers, which are also called the marker genes. And third, there should be cloning sites. And the fourth, there need to be vectors for cloning the plant and for cloning the genes in plants and animals. Now coming to the origin of replication. What do we understand by the origin of replication or ORI, as you can see in this picture? Now this particular is a sequence where replication starts. I hope you know what is replication, dear students. Can anybody of you tell me what is replication? Replication means the process of making DNA from DNA. That is called replication. सो डीएनए से डीएनए प्रोड्यूस करना जो है वो रेप्लिकेशन कहलाता है. राइट. सो इट इज़ अ सीक्वेंस वेयर रेप्लिकेशन विल स्टार्ट. मीन्स इट इज़ अ सीक्वेंस इन द ऑर्गेनिज्म फ्रॉम वेयर द रेप्लिकेशन प्रोसेस विल स्टार्ट. ओके. सो इफ एनी पीस ऑफ डीएनए, इफ वी लिंक एनी पीस ऑफ डीएनए विद द ओआरआई, देन इट विल ऑल्सो रेप्लिकेट विद द होस्ट सेल. तो अगर हमने ओआरआई पोजिशन पर कोई जीन लिंक्ड की है, so as the DNA will multiply, as the DNA of the host cell will multiply, that linked DNA will also get multiplied. Okay, I hope this is clear. Now in addition to this sequence is also responsible for controlling the number of copies of the linked DNA. जो ये ORI है, सबसे पहली चीज़ यहाँ पर रेप्लिकेशन स्टार्ट होता है. दूसरा, ये भी कंट्रोल करता है कि लिंक्ड डीएनए की कितनी कॉपीज बनाई जाएंगी. सो इफ वी वांट दैट देयर शुड बी लार्ज नंबर ऑफ कॉपीज ऑफ द टारगेट डीएनए, अगर हम चाहते हैं कि लार्ज डीएनए जो हमने इंसर्ट किया उसकी लार्ज कॉपीज बननी चाहिए, देन इन दिस कॉपी मस्ट बी इंसर्टेड इनटू अ वेक्टर. ओके. द सिलेक्टेड कॉपी ऑफ द जीन शुड बी क्लोन्ड इन अ वेक्टर हुज ओरिजिन सपोर्ट हाई नंबर. ठीक है. हमें ऐसे प्लाज्मिड के अंदर इंसर्ट करना है जिसका ओरिजिन ऑफ रेप्लिकेशन सपोर्ट करता है दैट इट शुड प्रोड्यूस लार्ज नंबर ऑफ कॉपीज. सो 3 things are very very important. ORI means it is a sequence where replication will start. Secondly, if you link any gene with the ORI, so when the host DNA will multiply, automatically your linked DNA will also multiply. Secondly, ORI also controls the number of copies to be formed for a linked DNA. So if we want large number of copies to be produced, we should link the DNA with a origin of replication that produces large number of copies. Second is your selectable markers or the marker genes. देखिए ये वो जीन्स हैं जो हमें ये आइडेंटिफाई करने में हेल्प करती हैं कि which cells are transformed and which are non-transformed. Transformed means जिन सेल के अंदर हमारा मॉडिफाइड डीएनए प्रेजेंट है, राइट. जो हमने नया डीएनए इंसर्ट किया है तो वो जिन सेल्स के अंदर वो प्रेजेंट है दैट इज कॉल्ड ट्रांसफॉर्मेंट्स, और जिन सेल्स के अंदर वो प्रेजेंट नहीं है दोज आर कॉल्ड नॉन ट्रांसफॉर्मेंट्स. ठीक है. सपोज यू हैव दीज सेल्स 1, 2. सो वी हैव इंसर्टेड सम फैक्टर हियर. Let's take different one. Let's suppose we have 2 cells like this and we have inserted some DNA in this particular cell and there is no DNA. You can say this here it is having its own DNA. तो यहाँ हमने recombinant DNA insert किया है. So selectable marker जो है वो identify करने में help करेगा कि ये जो cell है, this is the cell where we have inserted a foreign DNA, right? So that is why the selectable markers, they help us in identifying the transformant cells from the non-transformants. So transformation is basically a process in which a piece of DNA is introduced into the host bacterium. So ट्रांसफॉर्मेशन एक ऐसा प्रोसेस है जिसमें एक फॉरेन डीएनए को हम होस्ट बैक्टीरिया के अंदर इंट्रोड्यूस कर देते हैं. ओके, इज इट क्लियर एवरीवन? Like we talk about the selectable markers of E. coli. So there are certain genes which act as selectable markers in case of E. coli, that is ampicillin, chloramphenicol, tetracycline or kanamycin. These are the genes that will act as the selectable markers. Okay, if a normal cell is there, there is a normal bacterium E. coli is there, so it will have all the genes. It will have a gene which is responsible for producing resistance against ampicillin, chloramphenicol, tetracycline and kanamycin. Normal cells do not have any resistance against these antibiotics. जो normal cells हैं उनकी body में कोई antibiotic resistance नहीं होता है. ठीक है? Normal E. coli cells, they do not have any resistance against these antibiotics. Okay? I hope this is clear, everyone. Right, next comes the third point, which is called the cloning sites. Cloning site means the positions where the DNA, foreign DNA or the alien DNA can be linked. Right? तो ये वो positions हैं जहाँ आपका foreign DNA insert किया जाएगा. So those are referred to as the cloning sites. So if you want to link the DNA, if you want to insert the foreign DNA, then the vector needs very few restriction sites. तो क्लोनिंग जो आपका ऑर्गेनिज्म है, वैक्टर है, उसके अंदर few selected recognition sites must be there. Only, okay, if possible, very few recognition sites must be present. If there is more than one recognition site, right, then it will generate several fragments and that will complicate our process. तो उसके अंदर minimum recognition sites present होनी चाहिए जहाँ पर हम अपना DNA insert कर सकते हैं. राइट उससे ज़्यादा अगर आपके पास साइट्स मौजूद हैं then that process the process will be complicated. Okay, now suppose let's take an example. Suppose we insert a foreign DNA, okay, at a restriction site in one of the two antibiotic resistance genes. देखिए ये आपके सामने E cloning factor है. पहले तो हम let's understand the cloning sites. Let's come to the E. coli. Let's understand the structure so that we can talk about it. Now this is a structure of E. coli. It's a E. coli cloning vector PBR322. So ये आपका cloning vector है E. coli का. इसके अंदर restriction sites देखिए. There is HIND3, E. coli R1, BAM-H1, SAL1, PVU second, PST first and CLA1. ये सभी आपकी restriction sites हैं. Then there is an origin of replication. This is origin of replication. These all are your recognition sites. Okay. Then there are two genes which is MPR and TETR. Okay, MPAR and the TETR. These are two antibiotic resistance genes. ये जो हैं दो जीन्स हैं जो इस बैक्टीरिया को एंटीबायोटिक्स के अगेंस्ट रेजिस्टेंस डेवलप करती हैं. यानी अगर इस बैक्टीरिया को एम्पीसिलिन या टेट्रासाइक्लिन दो मीडियम्स में रखते हैं, सो देयर विल बी नो इफेक्ट. दिस बैक्टीरिया विल बी रेजिस्टेंट टू दीज एंटीबायोटिक्स. राइट? एंड देयर इज अ रोप. Okay, there's a rope, there's a gene which is called rope. Now this gene, it codes for the proteins which will be involved in the replication of plasmid. तो जब ये plasmid replicate करेगा उस टाइम पर जो प्रोटीन्स की रिक्वायरमेंट होगी वो रोप प्रोड्यूस करेगा. एक जीन है जो आपके प्लाज्मिड में प्रेजेंट है एंड दैट विल प्रोड्यूस द प्रोटीन्स विच विल बी रिक्वायर्ड फॉर द रेप्लिकेशन ऑफ द प्लाज्मिड. ओके, नाउ लेट्स अंडरस्टैंड द प्रीवियस स्लाइड. नाउ व्हाट वी हैव टू डू, वी हैव टू इंसर्ट अ डीएनए in any one of the two places, like या तो हमने वो जीन M में इंसर्ट करना है या टेट में इंसर्ट करना है. सो फॉरेन डीएनए को हमने दो साइट्स में से किसी एक साइट में इंट्रोड्यूस करना है, ईदर ऑन द एम्प और इन द टेट रीजन. राइट? आई होप दिस इज अंडरस्टूड. नाउ सपोज करिए अगर हम फॉरेन डीएनए को बैम H1 साइट पर इंट्रोड्यूस कर देते हैं, दिस इज योर बैम H1 साइट, राइट। सो एम्प रीजन है हमारा, टैट रीजन है हमारा, टैट रीजन में हमने बैम H1 पॉइंट पर हमने अपनी फॉरेन मटेरियल को इंट्रोड्यूस कर दिया है। अब ये जो रीजन है ये इस बैक्टीरिया को टेट्रासाइक्लिन के अगेंस्ट एंटीबायोटिक रेजिस्टेंट बनाता है। मीन्स दिस बैक्टीरिया कैन ईजिली फेस द टेट्रासाइक्लिन, ओके, इट विल नॉट डाई, it is resistant to these two antibiotics, okay. So now we have introduced a foreign DNA in the TAT region. So what will happen if there is a foreign DNA? Now this recombinant, the organisms where we have introduced a foreign DNA, they will be called as recombinant plasmids. तो अब हमारे पास दो प्लाज्मिड्स हैं, एक जिसमें हमने कोई इंट्रोडक्शन नहीं किया है, जिसके अंदर हमने कोई फॉरेन मटेरियल नहीं डाला है, नॉर्मल सेल है, आपका नॉर्मल प्लाज्मिड है, और दूसरा प्लाज्मिड है जिसके अंदर हमने एक जीन को इंट्रोड्यूस कर दिया है, किस रीजन में, बैम H1 रीजन में, ओके। सो these plasmids, they will be now referred to as recombinant plasmids. अभी जो recombinant plasmids हैं, they have lost the tetracycline resistance. जो नॉर्मल प्लाज्मिड था हमारा, जो नॉर्मल बैक्टीरियल सेल था, वो हमारा एम्पीसिलीन और टेट्रासाइक्लिन दोनों ही एंटीबायोटिक्स को फेस कर सकता था, दोनों के रेजिस्टेंट था। लेकिन जब हमने बैम रीजन में, ठीक है, बैम रीजन में एक फॉरेन डीएनए इंसर्ट कर दिया, सो अब ये बैक्टीरिया जो है ये टेट्रासाइक्लिन के रेजिस्टेंस नहीं रहा, ठीक है। इज इट क्लियर? नाउ हाउ टू आइडेंटिफाई दीज टू सेल्स। सपोज ये हमारा वो सेल है रिकॉम्बिनेंट, ये हमारा नॉन ट्रांसफॉर्म, ये हमारे नॉर्मल सेल्स हैं। तो इनको अब हम एक मीडियम में ग्रो करेंगे एंड दिस मीडियम कंटेन्स एम्पीसिलीन, ठीक है। दिस मीडियम कंटेन्स एम्पीसिलीन। कैन यू सी दिस पिक्चर क्लियरली? Yes, so now we have two types of bacteria. One, the normal bacteria which can produce, which are resistant to both the antibiotics, ampicillin and the tetracycline. And the transformed cells, the transformed plasmid, which is not resistant to tetracycline. Now both the cells we will grow on a medium which contains ampicillin, right? So both will, do you think both will grow? Just think of it, what will happen? What will happen? So in this medium, right, because these bacteria they were resistant, so both the bacteria they will grow on this medium. So दोनों तरीके के बैक्टीरिया इस पर ग्रो करेंगे. Now from this bacteria, now transfer these bacteria in a medium that contains tetracycline. अब इसे हम ट्रांसफर करते हैं ऐसे मीडियम में जहां आपका, so first they have been transferred to ampicillin resistant medium, so both the colonies will develop, the transformed cells also, non-transformed cells also. Now next portion we will transfer it to a medium that contains tetracycline. अब हमने सभी सेल्स को एक ऐसे मीडियम में ट्रांसफ़र कर दिया है जिसके अंदर टेट्रासाइक्लिन मौजूद है, राइट. सो व्हेन वी ट्रांसफर टू टेट्रासाइक्लिन वी विल ऑब्जर्व that the normal cells they are able to grow in this medium, but the transformed cells they are not able to grow in this medium, right. So your non-recombinants they will grow in ampicillin medium as well as tetracycline medium, but the new bacteria, the transformed bacteria, the recombinant plasmids, the recombinant bacteria, they can only grow in the ampicillin resistant, they can only grow in the medium which contain ampicillin. They will not be able to grow in a medium that contains tetracycline. Thus we can differentiate between the two cells, the recombinant and the non-recombinant cells. Okay, I hope this much is clear, everyone. Okay, now in this case basically that we have discussed just now, one antibiotic resistance gene, okay, in this particular example one antibiotic resistant genes help us to identify the transformed and non-transformed cells, or we can say the recombinant or the non-recombinant cells, right? So if there is inactivation of one gene, that will help us identification, right? But this process of selection or identification of recombinants due to the inactivation of antibiotics is a very tedious process. It's a very cumbersome process. ये जो तरीका है ये आइडेंटिफाई करने का कि सेल्स जो हैं वो रिकॉम्बिनेंट, कौन से सेल्स रिकॉम्बिनेंट है, कौन से सेल्स नॉन रिकॉम्बिनेंट है, it's a very tedious process. It's a very long process. And you have to, we have to plate 2 plates every time. Okay, we have to, you can say select 2 plates. We have to grow 2 plates having different antibiotics, which is a very, very, you can say, cumbersome process. So it was thought that we should use some alternative selective markers. We should always have some alternative selectable markers to differentiate between the recombinant and non-recombinant cells depending upon their ability to produce a certain color. So if they have a color, they are recombinants. That the presence of a color or absence of a color can be used to identify a transformed cell from the non-transformed cell. So if you look at the cell and from the color itself you can identify whether this cell is a normal cell or it is a transformed cell, right? So then this process becomes more simpler. As you can see here, suppose you can grow the bacteria here, and depending upon the color, like you can have blue color, red color, green color. So depending upon the color, you can easily identify whether they are combinants or the recombinants. Then coming to the vectors for cloning genes in plants and animals. Okay, there are certain, you can say, okay, let's have another example also. As I was telling you that if we have to identify the recombinants from the non-recombinant cells, so the easiest way is depending upon the chromogenic substrate and depending upon the color. Okay, now suppose we have a medium. Okay, suppose we have a recombinant DNA and we are inserting the recombinant DNA in a sequence that codes for enzyme beta galactosidase. सपोज करिए ये हमारा क्लोनिंग वेक्टर है, ये उसका डीएनए है, ठीक है। इसके डीएनए में हमने इस पोजीशन पर एक रिकॉम्बिनेंट डीएनए इंट्रोड्यूस कर दिया है। तो जहाँ पर हमने रिकॉम्बिनेंट डीएनए इंट्रोड्यूस किया है, दिस पोर्शन ऑफ दिस होस्ट डीएनए, इट प्रोड्यूसेस एन एंजाइम विच इज़ कॉल्ड बीटा गैलाक्सिडेज, ठीक है। एक सेल है, सेल के अंदर हमने एक फॉरेन डीएनए इंट्रोड्यूस कर दिया है, और कहाँ पर इंट्रोड्यूस किया है, वो एक ऐसी एरिया में इंट्रोड्यूस किया है, जिस पोर्शन में हमने इंट्रोड्यूस किया है वो पोर्शन एक एंजाइम प्रोड्यूस करता है जिसको हम कहते हैं बीटा गैलेक्टोसिडेज। ओके, so that gene or the portion of the DNA which codes for an enzyme beta galactosidase. So when we have inserted a DNA into this particular portion, so what will happen? There will be inactivation of the gene. तो जिस एरिया में हमने अपना फॉरेन डीएनए इंसर्ट किया है उसकी वजह से क्या होगा कि वो जो डीएनए है वो इनएक्टिवेट हो जाएगा एंड द एंजाइम विल नॉट बी प्रोड्यूस्ड। अब जो एंजाइम है वो प्रोड्यूस नहीं होगा, ठीक है, बिकॉज दैट पर्टिकुलर पोर्शन was coding an enzyme which is called galactose. But now because we have inserted a recombinant DNA, that portion of DNA will not produce the enzyme. Now this particular part is called the insertional inactivation. कि अगर insert करने से एक particular DNA के अंदर कुछ भी insert करने से वो अपना काम बंद कर देता है, it is not able to function well. So this particular insertion is called as insertional activation. Now because of this insertional activation, these colonies they will not produce any color. Okay, so अगर हमने ऐसा किया है तो जो normal DNA होता है, normal cells होते हैं वो colored होंगे और जो recombinant cells होंगे वो उनमें कोई कलर नहीं होगा, दे विल बी कलरलेस, राइट? क्लियर? सो इफ अ प्लास्मिड has no insert, then it gives blue color, and if the plasmid has an insert, then it will be transparent. तो अगर एक नॉर्मल सेल है जिसके अंदर बीटा गैलेक्सिडोज एंजाइम फंक्शन कर रहा है, सो उस केस में बैक्टीरिया का जो कलर है वो ब्लू कलर होगा. लेकिन अगर हमारा if we have introduced the DNA in this portion, so this will result in the inactivation of the beta galactidase, and such bacteria, they will be colorless. As you can see here, like this is your nutrient medium where all the bacteria is present. Then we have inserted some DNA. Okay, so these cells which are appearing blue, that means they are perfectly normal cells. There is no insert here, and the beta galactidase enzyme is properly functioning. But in this case, these cells are transformed, but they do not contain any recombinant DNA, right? But here they contains these cells, they have changed color, or they are transparent because the enzyme beta galactidase is non-functional here. Clear? So from the color itself we can identify. Now identification with the help of the color is a more simpler process, more easier process to identify a recombinant cell from a non-recombinant cell. Okay everyone? Now coming to the vectors for cloning genes in plants and animals. Now see students, you will be surprised that transferring genes into plants and animals from bacteria and viruses has been done from ages. हम बैक्टीरियाज और वायरसेस में से कुछ जीन्स निकालते हैं और उन्हें प्लांट्स और एनिमल्स में इंट्रोड्यूस कर देते हैं. ये कॉन्सेप्ट बहुत पुराना है. इट्स अ वेरी ओल्ड कॉन्सेप्ट, राइट? I hope you know it. Okay. So we will take out the genes from the bacteria and viruses and we will introduce them into plants and animals. Like for example, Agrobacterium tumefaciens. Okay. This is a pathogen in dicot plants. ये एक pathogen in dicot plants में. And this pathogen, it can deliver a piece of DNA which is called as tDNA. ठीक है? Agrobacterium tumefaciens. This is a pathogen in dicot plants, and this pathogen, it can deliver a piece of DNA. Okay, it can deliver a piece of DNA which is called tDNA into the normal cells, and once it is delivered into the normal cells, they will become tumorous cells. They will start producing a tumor. This means that tDNA will direct the normal cells to produce the tumor cells. And these tumor cells, they will produce a chemical that is required by this pathogen. So Agrobacterium tumescens क्या है? ये आपके you can say plants के अंदर एक DNA का पोर्शन इंसर्ट करता है जिसको हम tDNA कहते हैं. तो जब ये tDNA का पोर्शन इंट्रोड्यूस हो जाता है तो प्लांट सेल्स जो है वो नॉर्मल सेल्स से कैंसरस हो जाते हैं, ट्यूमर बनाने लगते हैं, और वो ट्यूमर वो केमिकल्स प्रोड्यूस करता है जो इस पैथोजन के लिए रिक्वायर्ड है, जो उसकी ग्रोथ के लिए रिक्वायर्ड है. Second is tumor inducing plasmid. Okay, tumor inducing plasmid. What do we call it as? Tumor inducing plasmid of Agrobacterium tumefaciens. ठीक है? This is a second example. Tumor inducing, that is TI, plasmid of Agrobacterium tumefaciens. ठीक है? इसको मॉडिफाई किया जाता है so that it is not pathogenic to the plants. Right? इसको मॉडिफाई किया जाता है, has now been modified into a cloning vector. So the tumor inducing plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plants, but it is able to use the mechanism to deliver the genes of interest into the plants. तो ये अब जो है ये पैथोजेनिक नहीं है, लेकिन ये आपकी जीन ऑफ इंटरेस्ट को आसानी से प्लांट्स के अंदर डिलीवर कर सकता है. सिमिलरली वी कैन ऑल्सो यूज रेट्रोवायरसेस. सिमिलरली जो है हम अपने रेट्रोवायरसेस को भी यूज कर सकते हैं. द डिसआर्म्ड रेट्रोवायरसेस मीन्स the retroviruses which does not cause any harm to the animal cells. Okay, they can also be used to deliver the specific genes into the animal cells. And once a gene or DNA has been inserted into a suitable vector, जैसे आपका जीन जो है वो insert हो जाएगा normal cells में, so that normal cells they will transform into the cancerous cells, so they can be used to deliver the desirable genes. तो रेट्रोवायरसेस जो हैं ये एनिमल सेल्स को नॉर्मल सेल्स से कैंसरस सेल्स में कन्वर्ट कर सकते हैं, एंड दे कैन ईजिली डिलीवर अ जीन और अ फ्रेगमेंट ऑफ डीएनए इनटू अ सूटेबल वैक्टर. ओके, वेयर दिस रिक्वायर्ड डीएनए कैन मल्टीप्लाई, right? It can multiply, whether it can be transferred into a bacterial cell or in a host cell. Okay, everyone? I hope this much is clear. Any doubt anybody has now? Okay. Now coming to the third concept, that is called competent host. Okay, that is for transformation with recombinant DNA. Now dear students, we know DNA is a hydrophilic molecule. Okay, it is a hydrophilic molecule, so it cannot pass through the cell membranes. DNA जो है आपका हाइड्रोफिलिक मॉलिक्यूल है, तो ये मेम्ब्रेन्स के थ्रू क्रॉस नहीं कर सकता है, राइट? देन हाउ वुड द DNA विल गो इनटू द सेल? So in order to force the bacterias, okay, in order to avoid this situation, or in order to force this DNA into the cell, the bacterial cells, they are treated with specific concentration of divalent cations. हम bacterial cells को divalent cation से treat करते हैं, like for example calcium से treat करते हैं, so that the DNA can easily pass through pores into the cell wall. जो कि डीएनए है वो आपका सेल पोर्स के थ्रू पास हो जाए, सेल वॉल को क्रॉस कर जाए. नाउ फॉर डूइंग दिस व्हाट वी डू, फर्स्ट यू कैन सी हियर, फर्स्ट ऑफ ऑल वी विल इंक्यूबेट द डीएनए, द रिकॉम्बिनेंट डीएनए ऑन द आइस. सबसे पहले हम बर्फ़ लेंगे, बर्फ़ के ऊपर हमारे रिकॉम्बिनेंट डीएनए को फ्रीज कराएंगे, राइट? देन इमीडिएटली आफ्टर दैट दे विल बी प्लेस्ड एट अ 42 डिग्री सेल्सियस टेम्परेचर. तो इन सेल्स को एकदम से शॉक लगेगा. ओके, पहले हमने उनको आइस ट्रीटमेंट दी है, नाउ वी आर पुटिंग द सेल्स डायरेक्टली ऑन दी 42 डिग्री सेल्सियस टेम्परेचर, सो वी आर गिविंग देम द हीट शॉक, राइट? एंड देन वी विल अगेन पुट देम इन द आइस. नाउ बिकॉज ऑफ दिस इमीडिएट शिफ्टिंग फ्रॉम लोअर टू हायर टेम्परेचर, the bacterias will take up the recombinant DNA. Okay, the bacterias they will be forced to take up the recombinant DNA. पहले हमने सेल को जो है आइस पर रखा, एकदम लो टेम्परेचर प्रोवाइड किया, फिर हमने उसे हीट शॉक दिया, एंड देन अगेन वी हैव शिफ्टेड द बैक्टीरियल सेल इनटू द at the 0 degree Celsius, that is on the ice. So because of this shifting temperature, the bacteria they will be forced to take up the recombinant DNA. Now apart from this, there are other several methods which can be used to deliver the foreign DNA into the host cells. Like first of all, we can have a micro injection. As you can see in the first picture, we can use a injection which is having a very fine needle, and the DNA can directly be introduced into the cell, into the nucleus of the host cell. So with the help of a micro injection, we can directly introduce the recombinant DNA into the nucleus of an animal cell. Second is called your gene gun or the ballistic process. इसमें the cells are bombarded with high velocity microparticles of gold and tungsten which are coated with the DNA. इसमें हम क्या करते हैं, as you can see this is a gun, so we will bombard the plant, we will bombard the plant with gold and tungsten particles, and these gold and tungsten particles they are coated with DNA. So when they are bombarded with a high speed, they get inserted into the plant. Now similarly, in the same way, we can also use, as I told you, the disarmed pathogen vectors. Disarmed means the vectors which cannot cause any disease. Pathogen vectors, जिन वैक्टर्स की वजह से हम ह्यूमन बॉडी में कोई ना कोई डिजीज कॉज होती है. सो वी हैव disarmed them, we have inactivated the pathogen vectors. So when they infect the cell, they will transfer the recombinant DNA into the host cell. So in totality, we can use three different methods like microinjection, we can use gene gun, we can also use the pathogen vectors, right? Is it clear? In addition to the shock treatment, that is heat shock, like we are putting once the DNA, recombinant DNA on the eyes, then we put it on the 42 degrees Celsius, then again shifting back to the eyes. So we have basically 4 methods of introducing the recombinant DNA into the host cell. Okay, everyone? Oh, you can discuss your queries if you have any. Any doubt, anybody? Okay, so coming to the homework for today. First of all, you will make notes of this topic. It's a very, very interesting and a very useful topic. Secondly, you will read the page numbers 198 to 200, and if there is any doubt, any query you can discuss it with me on my personal number. Dear students, while reading through the book, there are certain words which are highlighted with black. So they are very, very important. So when you are reading, do try making the small questions, 1-mark questions, and understand the meaning of those black highlighted words. And even then, if there is any doubt, anything that you need to discuss, you can call me. Okay? Okay, so that's all for today. Bye, everyone.